Café. My name is Helen Lewin. I'm the Program and Partnerships Manager at Cary Library. Before we begin, I want to take a moment to thank the Cary Library Foundation. Their support enables us to bring programs like today's event to you. The foundation plays a unique role with the Science Cafe series and produces the content for these programs. I'd also like to thank the public libraries in Ashland, Bedford, Needham, and Quincy for partnering with us on this program. This program is being recorded and will be posted to the library's YouTube channel. We are running this program in a hybrid format. We will have time for questions at the end of our program. If you are joining us in person, please raise your hand and I'll bring over a microphone. And if you're joining us online, please submit your questions to the Q&A. Jagesh Shah will be your host for the evening. Jagesh is a distinguished scientist with a background in biology, computation, and engineering. He's also a Lexington resident, former Cary Library Foundation board member, and the creator of the Science Cafe series. Welcome, Jagesh. I'm going to pass the evening off to you to host and introduce our special guest this evening.
Thank you, Helen, and thank you everybody online, and thank you everybody here. We have a fairly large group today. I was telling Professor Langer that we don't usually have to put the seats in the back, but I think his as a distinguished guest, I think it makes a lot of sense. So welcome everybody to the Cary Memorial Science Cafe. This is something that I realized after looking at the calendar we've been doing for 10 years. Corin can tell me otherwise, but and the goal was to bring scientists into the library, talk about what their upbringing, how they got to where they are, and what they're doing now. And it's a chance for all of you guys to see, what it takes to kind of become a scientist, and also what the exciting things are that they're working on. So today we have a real treat. Professor Robert Langer is one of the world's leading inventors and is considered the father of modern drug delivery. So I think, you guys are gonna learn a lot about many of the inventions that I think you'll find that if you yourself don't take them, there's a good chance that somebody is using them regularly. Any of you that had a COVID vaccine, some of the materials that went into the Moderna or BioNTech are derived from the pioneering work of Dr. Langer. At MIT, he's an institute professor, and this is a prestigious honor bestowed on very few and really underscores a good deal of his many accomplishments. And because we want to hear from Bob, I'm not going to go through his long list of accomplishments, but I will have a small little vignette here. So he's received, I think 42 is the last number, honorary doctorates, 220 awards, including the U.S. National Medals of Science and Technology and Innovation, and only one of 3 living individuals to have had both honors bestowed on him. He's also a member of, I guess, the quadfecta of National Academies of Science, Medicine, Engineering, and Inventors. So we're going to walk through Professor Langer's journey. We'll start at the beginning, but I think, again, I really want you guys to really absorb this opportunity. We don't get a chance to always talk to somebody like Bob, and I think it's going to be a lot of fun. So thank you for being here, Bob. And maybe we usually start at the beginning, and usually your beginnings. And maybe, I think we'll find out you've had so many aspects of your career impinge on science, technology, inventing, but were there any things that happened early in your life that kind of gave rise to that?
Well, I'm not sure. When I was a little boy, and I'm old enough, some of the older people in the audience may remember this. Well, some of the presents I got, there was a company called Gilbert, and they made erector sets where you could build things like robots or rocket launchers. And they also made microscope sets where you could watch shrimp eggs hatch. And they made chemistry sets where you could, and I've always liked magic, where you could mix chemicals together and they changed colors. And all those things, I think, had some, I like to think maybe had some effect on me.
So I think when somebody chooses to be an engineer, it's a unique choice. It's at that interface of science and technology and application. So, what was it about your early kind of upbringing that, you chose to be an engineer, and when did you kind of feel like that was your path?
Yeah, well, that's an interesting question for which I'll be very embarrassed to give the answer. But, when I was in high school, I was good in math and science, and I was not very good in anything else, like French or English and stuff like that. So my guidance counselor and my dad said, well, if that's what you're good at and what you're not good at, you should be an engineer. And I remember thinking to myself, I really, this is true. I thought engineers ran railroad cars. And I thought, why would this make a difference? But anyhow, I didn't know any better, and I just felt, okay, I'll be an engineer. So,
Well, I think that, I hope that, our guidance counselors are also doing an equally good job of spotting talent at LHS. Somebody did a good job there. So one of the things that happens particularly in engineering is it's a discipline where many people go to college, and then they can get a job. It can be a professional degree where you can get a job. And I always like to point out that not only did most of the people that we bring into the Science Cafe go to school, but then they go to extra school. We call it graduate school. And, making that choice to keep on moving towards either more specialization. What was that? And tell us a little bit about your undergraduate
yeah.
experience.
Well, so at any rate, so I went to Cornell as an undergraduate. And actually, my first year, you had to make a decision what kind of engineer you're going to be. And the only class I liked and was good at was chemistry. And I did terrible in physics and things like that. So I said, well, I'll have to be a chemical engineer. So I did that. But I did fair in Cornell, but it was hard for me. But when I got done, or as I got to be a senior, some people got job offers, and I did. I got a couple of job offers to run chemical plants in like North Dakota or places like that. But I also felt like if I was running a chemical plant, I felt bad for the people working there. And so I thought, well, I didn't know what I wanted to do. I applied to graduate school. Also, that was part of the years when the Vietnam War was going on too. And at any rate, I was not excited about going into that either. But at any rate, I applied to graduate school. I went to MIT. When I went there, I still really kept not knowing exactly what I wanted to do. MIT, one of the things I liked, it was very diverse. You could have, do polymers, you could do bio, you could do batteries, all kinds of things. Still, what I did, I started doing this thesis, and it was okay. But what I got really interested in, I loved.
Talking 1 second to point the microphone at, because they can't hear you in the back.
Oh, okay.
Okay, there you go.
Thank you. I'm so sorry. I'm actually not a very good engineer. So I always say, like, my wife, even though she majored in neuroscience, she's a much better engineer. Like, if we need a light bulb changed, she'll do a much better job than I will. Actually, almost anybody would. But at any rate, so when I was a graduate student, first when I was an undergrad, I loved being a teaching assistant. And so when I was a graduate student, I got involved in helping start a school for working class kids in Cambridge. It's called the Group School. And I loved that. I spent most of my time as a graduate student doing that, or at least for a couple of years. And I wasn't really excited about the research I was doing. I tried to get, I'll let you, I probably shouldn't spend that much time on graduate, but that's basically what I was doing. And I guess when I got done, all my friends would go to oil companies. So I thought, I'll go to an oil company. They had so many offers. In fact, again, for those of you that are old enough, if you lived in Cambridge in the 1973 or 4, and you had a car and you went to a gas station, you'd have to wait in line for 2 hours to get your tank filled up. It's hard to believe that today, but that's true. The consequence of that, though, is if you were a chemical engineer, you got a lot of job offers. And they paid tremendous amounts of money. So I thought, I'll do what everybody else does. I'll work, and by the way, I had a pretty sheltered life. I was 25 years old. I had not been west of Buffalo or east of Boston, but at any rate, now for the first time in my life, I get to see the country. I have all these interviews, and I got 20 job offers from different oil companies and chemical companies, 4 from Exxon alone. And I wasn't that good, but they just had so many openings. So at any rate, I just decided I wasn't gonna do that. One of the guys, I still remember, they got older MIT people to meet you and interview you. One of the people at one of the Exxon said, gee, if you could increase the yield of this one petrochemical by 0.1%, he said, that would be fantastic. It's worth billions of dollars. And I remember flying back to Boston thinking that I didn't want to spend my life doing that. So I did. One day, and since I like teaching so much, one day I saw an ad to develop chemistry curricula. That's one of the things I started doing at this school at the City College of New York. So I wrote them a letter applying for a job. They didn't write me back. But I really liked that idea. So I found all the ads I could to do something like that. I found about 40, and they weren't even in very good colleges. Nonetheless, I don't think any of them wrote me back. So that wasn't, so I had a hard time finding what I want to do. I started to think of other ways I could use my chemical engineering background to help people, and I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. And then one day I walked into the lab where I was, and one of the older scientists said, he said, Bob, he said, there's a surgeon in Boston named Judah Folk. And he said, sometimes he hires unusual people. I mean, he thought very highly of Dr. Folkman. I won't say what he thought about me. So that was kind of my post high school career.
Well, I think this is exactly, kind of the story that, I don't know how many of you expected to hear kind of this nonlinear path, but many of, the people that we have in these vaunted positions, they have a good nose for what they want to do, although it might take them a while to get there. But I think that, you introduced now Dr. Folkman, who brilliant surgeon, a remarkable scientist. And, you spent time in his lab, and this was, I gather, that transition into bringing the chemical engineering know-how that you had into medicine.
Yeah, well, that job, I mean, I was the only engineer in the entire hospital. And it was, I mean, on the one hand, it was tough because I didn't know any biology. I mean, still, the last time I took a biology course or anything like it was in 10th grade. And of course, that's what you were doing, with the goal that he had a theory, for those of you, and that's probably most of the people, he had this idea that if you could stop blood vessels from growing, that might be a whole new way of treating cancer, possibly other diseases. And it turns out that he was widely criticized for that. People didn't think that was possible or that he was right. And of course, what did I know? I mean, I thought it sounded like a pretty good idea to me. And also it struck me as important, that if you could really do that, then maybe you really could make a difference someday in people's lives. So my job, he said, was to prove he was right. And in so doing, isolate the very first blood vessel inhibitors. I also got to see a lot of the hospital. I made a lot of friends who were clinicians, and I learned a tremendous amount. It was also hard. I was trying to understand biology and, materials and all kinds of things. And I had to keep reading and, and it was frustrating too, but it was a great experience.
Yeah, I think, making those transitions from one discipline to the other does require your kind of, that it's that curiosity that you brought and being able to keep at it. So, again, this is, in the academic speak, this is what we call a postdoctoral fellowship. So for those of you that are still counting, we're still kind of in school. So I can say that because I did.
Yeah, and by the way, it's an interesting thing, then being an engineer, almost nobody did postdoctoral fellowships. You know, you could get jobs in oil companies or in academia, you know, right away. But, you know, I didn't even think of it that way. I just thought it seemed more important to me than any of the other things that I was thinking about.
And so, you know, that early introduction to clinical medicine is something that most engineers will never have had experience in, even to this day, to see clinical workflow, to be able to be in a lab where you're bringing this interdisciplinary. It's maybe more common now, but certainly back then you would have been one of the pioneers. And you know, once this fellowship was, you know, at some point complete, there was a transition where you decided to go into academia. And what was that transition like? And what were the first few years of that experience like?
Yeah, well, let me go back a little bit to the hospital. So to isolate angiogenesis inhibitors, there's no way to study blood vessels in, so we had to invent one. And it's not so easy because there's very few places that don't have blood vessels. So you don't want a lot of background blood vessels. Two of such places are the cornea and the choroidal or membrane. So our idea was you put certain type tumors in, and then we needed to have, and also I was isolating these different molecules, all of which were what we call really large molecules. And for reference, proteins are large molecules. Nucleic acids like DNA and RNA are large molecules. And so what we wanted to do was to take some of these things that we isolated and have a delivery system, but it had to be very tiny to put in these places, like the choroidal membrane or the cornea. So like a nanoparticle or a microparticle. So what we needed to do was to be able to put these large molecules in these tiny particles, nanoparticles, and deliver them. So at this point in time, 1974, there was pretty much zero work going on in drug delivery. There's only one company in the world, Alza, that was working in this area. Dr. Folkman was on their board, and they had Nobel Prize winners, famous people on their board, and he asked them, could they help? And they said, well, no, you know, it's just not possible to deliver large molecules, kind of like if none of us could walk through a wall like that. So they told us it was impossible, but if we were going to solve the blood vessel problem, we had to solve this problem. So I spent several years working on this. I actually found several hundred ways to get it to not work. But eventually I did. And so we published two papers. I remember one in Nature, which was the first delivery of these large molecules from tiny particles, and one in Science, using that to actually show that blood vessel inhibitors did exist and provide a pathway for future inhibitors to be found and discovered. So, you know, and I really was excited about this work. And I love being a postdoc. I probably might still be a postdoc, but a lot of my friends said, you know, probably not so smart to spend your whole life doing that. And I kind of agreed with them. So I started applying for faculty positions now in chemical engineering, which was my background. And no chemical engineering department in the world would hire me. They all said this bio stuff doesn't make any sense. I also tried to get research grants. My first 9 got rejected. They were generally reviewed by what are called study sections, and from the National Institutes of Health, like their pathology study sections. And they would say things like, well, Dr. Langer is an engineer. He doesn't know anything about biology and even less about cancer, which I thought was hard to know less than nothing. But you know, I had some math background. And so but it was pretty rough. So what happened was ultimately, since I couldn't get any jobs in any of those places, Dr. Folkman introduced me to Nevin Scrimshaw, who was head of MIT's nutrition department. And again, he was also a very, very famous nutritionist, was an institute professor. But he was kind of what I guess I'd call a benevolent dictator kind of department head. He liked me, so he offered me a job, but he didn't ask anybody else in the department what they thought, which would have been okay, except for one thing, which was the year after I joined the department, he left. And so a lot of the faculty decided to give me advice, and their advice is I should leave too. So I didn't have a great beginning at MIT either.
So we know how it turns out, right? So.
You have
This
to,
is
you have
the-
to hang in there, right?
Yeah, this is like the dramatic part of the movie, right? Where you're like, oh no, the hero's in trouble. Something bad is gonna happen, but I think that again, I don't know if this is one of the themes here is this idea of curiosity and perseverance, right? You stayed in the department. My understanding that this department doesn't exist at MIT anymore.
Yeah, that's a whole other story, but you're right.
But this early foray into the microparticles and the nanoparticles, and more importantly, the materials that went into making these were some of the earliest versions of what we would call drug delivery. And, at some point, there's a inflection point where now you're putting drugs into these and these are going to go into people. And that was an early part of your career that you got to make that transition, because I think anybody who's been in the industry, the ability to be able to say one day that you put something that actually went into a human being and changed their lives is kind of a remarkable experience.
Yeah, well, what happened there too, so I kept working on things and you're right, I was excited about the science and we kept doing science and I had a lot of very good students, some of whom were in the audience and lots of others. But, I always had this dream, I guess, that right along the lines you said, I wanted to make a difference and with the work we did. So it took about 10 years before anybody seemed to care. I mean, people would cite our work, in references, but I really wanted to see things get to people. And I was very naive. I think lots of people were then, but I was very naive. I thought if I wrote articles, then companies would use it, things would happen. But it didn't happen. And so so what I started doing was writing patents. And finally, after 10 years, we got the patents rejected many, many times. That's a whole other story. But finally, we got them because I found a quote that was very much along the lines that I said before, that this was impossible. And when we showed that to the examiner, he allowed the patent because we showed that it was. But at any rate, so after 10 years, I get these calls from a couple of multi-billion-dollar companies. One was International Minerals and Chemicals. They were interested in delivering certain molecules to animals. The other was actually Eli Lilly, and they were interested in delivering insulin to people. And I thought, and they gave me like a consulting fee. They gave me a grant, which for a young professor was very important. Most important and exciting to me was they were going to develop this and get it to patients. And what happened is they worked on it maybe for 6 months to a year, but they didn't get exactly the results they liked, so they gave up. And I remember about 2 years later, Alex Klebanoff, one of my friends, said, Bob, we should start our own company. And so we did. And I've kept doing that ever since. That was 1985. Since that time, we've started some 40 companies. We've licensed things to probably over 400 companies. And so but it's really, and a lot of my students, of course, worked at a lot of these companies, but I think it's that interface between what we did academically and developing the principles and the patents and then all these people at the companies really doing this great work, maybe building on what we did, but taking it so much further, that enable, products that, affect an awful lot of people.
Yeah, and I think this is probably one of the most exciting transitions is to say, sometimes certainly I can speak for myself when, I became a professor, my mother was so proud. Oh, you're a professor. That's good. And what you saw was that you would write these papers but your instinct was that, nobody's doing anything with these things that I'm trying to be an engineer. This is kind of the heart of an engineer is that application is to change something and take technology and put it out there. So you decided to do something which was start your own company, which anybody who's worked in academia is hard enough to be a professor, let alone start a bunch of companies. So what was the motivation came from really feeling like you needed to have that technology and that you wanted to be a part of that process?
Well, I'd say that that's pretty much right, but what I felt, like you said, why did I do all this in the first place? The teaching, the research, I wanted to see it help people. And I thought the research was pretty good, I could see this with my own eyes, the delivery work, and actually the blood vessel work that we did would later lead to drugs like Avastin and Eylea and Lucentis, drugs that are also used by many, many millions of people. I mean, that was Genentech and other companies' work, but it built on that initial discovery. But, of course, I didn't know that then. This was now the 1980s. That didn't come till the 2000s. But I wanted to see it make a difference. And I kept trying to think to myself, you can only go so far at MIT. MIT is a wonderful place. And we did, I think, really what I like to think is a seminal work. But to really make that difference, would need to go further. You can't do manufacturing at MIT. You can't do clinical trials at MIT. You can't do really a lot of development. So I thought, companies, and in fact, those two large companies, Eli Lilly and International Minerals and Chemicals, could do it. But the problem is a lot of times with large companies, people can give up too easily. And even if there's champions there might be some other person higher above them that would say, well, even if you think this is important, we don't, and then it doesn't happen. So, so slowly I could start to see that we could develop either relationships with companies or start our own companies. And I could see when we did the little companies that they would make a difference and did make a huge difference.
Yeah, and I would say that I don't know what the ecosystem at MIT was like then, but now it is kind of a remarkable place for entrepreneurship. Now it's a place where one of my conversations recently with the former provost, Marty Schmidt, said he was talking to a young faculty member and she was really getting grants and doing well and publishing, but she looked kind of upset. She said, I haven't started my first company yet. And it really is kind of in the ethos of a place like MIT to be able to get these. And I think, Bob represents one of these forefront, people in the forefront that really developed that ethos. So why don't we get into, I thought maybe we could get into something that I know that we've discussed at a previous Science Cafe. For those of you that might have been with us during COVID, when all of these were fully virtual, we got a chance to talk to some Lexington residents, one who was Undersecretary of the Housing and Transportation, who ran the COVID task force. And then we got to talk to Melanie Iverson, who was the Chief Development Officer at Moderna. And in both of those cases, we heard a little bit about the industry and the government pieces. But I thought, maybe Bob is also one of the founders of Moderna, and part because the technologies that he brought would help us encapsulate what he called that large molecule, which in this case was messenger RNA that would code for the vaccine. And so what was that experience like? I think, you're now, undoubtedly you've helped a lot of people with many of your companies, but this was going to be on a scale that was rather different.
Yeah, well, going back to Moderna, so, we kept now, I think some of this work, I was in the 1980s, we kept doing this, finding out more in the 1990s, 2000s, and so forth. Well, to tell you the RNA story, so if you look at what happened historically, the first really key paper written by a guy named Wolf and some others came out in 1990, where they showed that RNA could make proteins in animals. And there were some papers after that, but what would happen is sometimes the RNA would have some not so good effects. In 2005, Drew Weissman and Caitlin Carrico made an important finding that they could modify the RNA a little bit and stop that from happening. They wrote a patent and they started a company, but nobody would fund them. I should say, well, anyhow, then, by the way, in the process of that, one of the other companies I got involved in being on the founding scientific advisory board was a company called Alnylam. Alnylam, we were trying to make drugs out of the opposite of messenger RNA, of siRNA, which could knock genes down. And I got one of my students, Kenneth King, he was one of their first employees. And Phil Sharp, who's a Nobel Prize winner, who was one of the founders of Alnylam, he always said, well, the biggest issue in genetic therapies is, as he put it, is the delivery problem. So at any rate, I think a lot of investors, I think, just naturally assumed that would never get solved. So, and they didn't. So even though Caitlin and Drew did amazing work, nobody would fund them. So about 4 or 5 years later, Derek Rossi, who was a young professor at Harvard, he and his team built upon that work, and he came up with a way to change what's called a fibroblast, a cell, into what's called an iPS cell, which is kind of like a stem cell. And he did it by modifying the RNA the way they did. And he wanted to start a company. So he went to Third Rock, which is a great place. And they said, again, we're not going to fund you. And Tim Springer said, well, maybe he should come over and talk to me. So he showed me, he had like 3 main ideas. One might be that you could use it for like things like stem cells. Another might be kits. And a third might be to make proteins. And I thought to myself, we could certainly do this. We, over the last, now from 1974 to 2010, some of the things in our lab, well, first we showed you could deliver things like RNA when people said you couldn't. But then we actually came up with certain coatings that are called PEG, polyethylene glycol. In fact, Jeff Herkash this year was involved in some of that. And that would prevent the little particles from sticking together and also from being eaten by certain other cells. Then we came up with a couple of other of my postdocs, Dan Pack and David Putnam, came up with a way to make a certain type of thing which would be used for what are called now ionizable lipids, so that none of these things would cause toxicity. And then Dan Anderson, another postdoc of mine, now professor at MIT, came up with a way to make many libraries of these, many ones. And finally, Rohit Karnak, another postdoc of mine, in 2008, came up with ways to make these by what are called microfluidics, which to me seemed like a scalable method of manufacturing. So I thought to myself when Derek came to see me, well, we could solve this problem. I introduced him and Tim to Nubar Fagan, who I was on Nubar's thesis committee, and Nubar is really a visionary, and he said, well, you'd like to be involved with us and help provide some initial funding. We also got Ken Chen, who was a cardiologist, involved. So that's how it started. I think really to one of the points we talked about before, there's only, no large company, no large pharmaceutical company, for whatever people tell you, would touch messenger RNA. And so there were only 2 really companies that did for many years, and that was Moderna in the US, and then BioNTech in Germany, also CureVac in Germany. It wasn't until 2020, 10 years later, that Pfizer got involved by making an arrangement with BioNTech to produce, the nanoparticles. But that's how it got started. I should say that when we started the company, it got ridiculed. Stock analysts, scientists, clinicians. I mean, the media. There's this journal Stat that they kept ridiculing it over and over again, saying it was overvalued at $2 billion. I think it's $60 billion now. But Boston Globe wrote nasty articles on it. So it was kind of sad to me to see this. But I think the people at Moderna, some of whom again were my students, and other students at like MIT and Harvard, they did an amazing job. And obviously, that would lead to the vaccine. And I think that I'm prejudiced, but I think that's just the tip of the iceberg.
I think that the tip of the iceberg analogy is truly appropriate. This lipid nanoparticle, there are many nanoparticles that have been developed. The one that's in these vaccines is just even in terms of the diversity of chemistry, still the tip of the iceberg. There's many opportunities, but the ability to be able to take the language that cells use to make proteins and deliver that same molecule to cells so that they will make them is kind of a remarkable time in medicine. It really does change our ability to think about translation. If you understand how the cell is talking, you can take that same language and treat a disease cell. You can make a cell do something that it didn't do before, or help it with the fact that it doesn't have the right complement of genes. So I think this genetic medicine, its delivery, now many people in the early days, and I think there are probably many people in this audience were ridiculed. When we used to say stuff like, "Hey, we're going to make the FedEx." They're like, "FedEx? You're not going to make any money doing that." FedEx makes a lot of money, by the way, but the FedEx of the body and being able to deliver these medicines inside the body is going to be kind of a transformational time in medicine. And the vaccine is just the first step. I think we're now seeing many opportunities for this kind of work. So maybe that's where you can take us in the last little bit, Bob, is the future.
Sure.
This is just what is under the water in this iceberg. What do you see as the opportunities?
Yeah, well, I'll mention a few. I probably should have made one thing clear. You might say, well, why do you need the nanoparticle in the first place? But if you didn't have it, the body would just destroy RNA just like that. So you need to protect it and deliver it, as you were saying, to the body and to the cell. So what's happening? So if I look at Moderna, for example, there's going to be a lot of other vaccines, like for RSV. I think one of the really exciting ones is what I'll call personalized cancer vaccines that you can actually, and this is now in advanced clinical trials for melanoma and other treatments where you could make a lot of vaccines are what we'll call prophylactic, right? You give it to the patient before they have the disease. That's what's true for COVID, for flu. Cancer vaccine is what's called the therapeutic vaccine. Here, what Moderna and Merck are doing in partnership is to take a patient's cancer cells and analyze them and take the mutations, which are called neoepitopes. So you take the top 34 mutations, neoepitopes, and now you make mRNA that will make a vaccine to destroy each of those. And you give it to the patient. And that's now, a lot of this has been announced, but and now it's in the final stages of clinical trials, what's called phase 3 trials. But they've been able to show tremendously increased survival when they do this. And of course, hopefully that will translate to other cancers as well, and to all kinds of things. Some of the other things, though, that people are looking at. In fact, I'll maybe embarrass Dr. Chapelenik, who lives in Lexington, who collaborates with me on a lot of things. Bill Gates came to see me, must have been about 12 years ago, and he asked, well, could we ever make a vaccine, that you could just give once, for people in Africa? This was way before COVID happened. And could you come up with a way to make a self-boosting vaccine? So Dr. Chapelenik has created a series of little capsules that can, like a little eggshells, that can break at different times. So one could break at time 0, one could break at a month, 2 months, say 6 months, whatever time you want, and you make a cocktail of them. So you give one injection, and again, this is in the research stage, but you give one injection, and the idea is you get, many pulses, and so you could think of that as a self-boosting vaccine. And she and another one of my former students, Mark Prousnes at Georgia Tech, they're putting them into little microneedle patches that are, and for microneedles, people may not have seen these, but I'm sure you've all seen needles. Microneedles are tiny. And so if you put up like a little Band-Aid with them on, you'll barely see them, but they'll prick you. It won't hurt very much because they don't go in very deep, but deep enough so that you can inject little particles like this. And the idea is that someday, hopefully, this or something like this, they could be shipped all over the world to, whether it's Africa. In fact, Dr. Chapelenik also created Barda came to see her, and she's made like a tiny little 3D printer that you can make these microneedles and again ship them to remote regions. other things that we're working on, with Dan Anderson, we're working on, who I mentioned, we're working on ways of targeting little particles to hopefully every cell in the body. We've started a company called Souffle Therapeutics to do some of this. And we're working on various things in the laboratory itself. That's just with RNA. I mean, there's so many other things that we're looking at. We've worked on ways of trying to even deliver things like this orally. Again, with Bill Gates, we came up with a way with GeoTraverso to make little pill capsules that you can swallow, but instead of lasting for a day at maximum, they could last for a week or a month. And we started a little company called Lindra, which is now in advanced clinical trials for diseases like schizophrenia, and also helping, we're working on diseases like malaria too. I could go on and on. We have a lot of stuff going on.
Well, I think maybe we'll just, we want to start the Q&A and give you guys a chance to ask some questions, but I did want to just, one of the areas, the interfaces that I think, your lab is working on and a number of people who have graduated from your lab is also not just the ability to deliver mRNA to be able to give a gene transiently, but also modify the gene, modify the genome. We've heard about CRISPR, and we've heard about the ability to modify the genome. We've heard about, for those of you that may have followed, the ability to now take cells out of the body, modify them, and then put them back in the body, and as a cure for sickle cell. And, how do you see, this is a tricky subject. I mean, modifying the genome inside the body, but there are remarkable opportunities for changing the lives of not just people with, chronic disease, but genetic diseases with these gene editing technologies. And that's really the convergence now of modifying the genome with delivery and, many of the diseases that I'm sure that you thought about your entire career.
Sure. Well, I mean, that's a whole other area that Dan and I have worked on that. Of course, a lot of the same tools will be applicable, but a lot of times I think gene editing from a delivery, if to do it outside the body is, certainly possible. Doing it inside the body, that's tougher. And that's again where the delivery nanoparticles are important, and where things that I mentioned, like targeting, like say for a vaccine, you don't need to target them, but for a lot of things, or to even make a new protein, you don't need to target them, but probably for a lot of the gene editing things, you do. And so we and a lot of our students and a lot of our former students are working on that, and a lot of companies too.
Yeah, and I think that, although, Bob was somewhat modest about this, he runs the largest biomedical engineering laboratory in the world. And I think there must be no port that you can go to where you don't have some trainee. And I think based on what I know, Bob, he usually checks in on them almost everywhere. He certainly is somebody who you will know because his support is long lasting. He looks after many of his trainees, and I can say even people that he might have met later in his career, he's looked after them too. So he's somebody who has spread not just technology, but also the wealth of his, kind of support. And I've benefited personally from that. So I thank you. I thank you here. But I know there's many people in the audience that are going to have questions, Bob. So thank you for being here, and thank you for your support, and thank you for bringing this to us.
Oh, it's my.
I think Helen's going to handle the.
Yes, so if
Q&A.
anyone has questions, please raise your hand. Oh, it's one first.
So one of the things about RNA delivery that I haven't seen emphasized a lot, and I'm in the pharma industry. RNA delivery, in essence, takes that chunk of money that we spend to develop manufacturing, takes it out of the equation, or takes it out of the equation because we are the manufacturer.
Exactly.
We're the manufacturing factory for this thing. Do you think, or can you what do you think is going to be the economic impact of essentially greatly simplifying manufacturing in terms of, say, less developed countries, and perhaps LMIC countries that are modestly developed, that have the ability, for example, to make generics, for them to ultimately have this technology translated so that they can develop drugs or whatever, vaccines, probably vaccines first, that are relevant to their particular needs. I can't imagine somebody making a vaccine in our place, in our developed world, for something like Chagas disease, for example. But why not spread the technology in such a way that those countries that suffer from that can, in essence, carry out programs, meaningful programs, take care of their population?
Sure. I think that's a great question. Let me take a step back for people that may not know as much as you do about the difference. So one of the things, and this gets to why I thought about this back in 2010. So I had been an advisor to Genentech, which is a pioneering company in protein therapeutics for probably over 30 years. Well, it was for over 30 years. But the way people classically made proteins, and by the way, I think something like 70% of the top best-selling drugs now are proteins, like things like antibodies that you read about. But classically, when people make large molecular weight drugs, they have to grow them in giant cell vats, or let's say, and I'm sure people are aware of this, when you're making vaccines, like say the flu vaccine, you grow them in eggs. You actually have to start maybe more than a year in advance. So not only does it take, is it very expensive, it's also very time consuming. And that time, of course, going to flu vaccines will clearly cost lives. I mean, if you think about it, right, when people make flu vaccine, they're always guessing, because you have to start maybe a year or two in advance. Is it H1N1, H1N2? They don't know. So they make their best guess. They're often wrong. If people, I remember being shocked myself a number of years ago when I got flu. And I remember talking to the doctor and I said, well, I got a shot. He said, yeah, but it's like 40% effective. And for exactly the reason I told you, they guessed. And so anyhow, the beauty of messenger RNA, and that's why when Derek came to see me, I thought about this immediately. If you could take sRNA, put it in a nanoparticle to protect it, inject it into the body, rather than the eggs or this giant cell vat being the factory, the body's the factory. That's really the gist, or the first part of his question. And I think it could do exactly what you said because of that. In other words, because the body's the factory, I think it's pretty unlimited in terms of what you can do. You can do it much faster. That's why Moderna and BioNTech, people may not realize this, the only two vaccines in the United States that ended up being really used, the Moderna vaccine and the BioNTech Pfizer vaccine. Two that succeeded. There are another 49 that failed. People don't hear about the failures, but anyhow, and these happen quickly, relatively speaking. So I think your point's right that you can hopefully treat all kinds of diseases in various countries, ours and other countries. I think what gets done, of course, is to publish things so that people can use them. What's also gets done, I mean, in fact, our lab's done an awful lot of this, is work with foundations like the Gates Foundation to try to, come up with therapies based on this. And companies, of course, only can happen so fast, set up plants in different countries or work with different countries to hopefully enable this to affect more people. And even like I was mentioning with Dr. Jackalenich's work, maybe come up with things that that you could someday give to these countries, like these 3D printers or patches that could be shipped. I didn't even mention that some of the things that Dr. Jackalenich did was to stabilize the mRNA vaccine, which we put in our Nature Biotech paper. But that's really key because they're not shelf stable. So anyhow, the hope is that all the things you say someday will come to pass. Of course, it doesn't happen overnight. It's a lot of work by a lot of people.
Thank you.
Great question.
So I have a question. The work that your work, has it been applied to any of the brain diseases like Parkinson's or dementia or Alzheimer's?
Well, it's also a great question. You know, it's funny, about a year ago, I got asked to speak to the board of directors of GlaxoSmithKline. And the chairman of it asked me almost exactly that question. He said, how come Moderna could make this vaccine in less than a year and get used in lots of people? And we've been working for 30 or 40 years on Alzheimer's and we have almost nothing. Well, my answer, and this is really relates to something that Jaggesh was saying, is understanding the biology. You know, we knew exactly what the problem was, you know, with you go after the spike protein in COVID. That was scientifically understood. I think we still, unfortunately, don't know enough about brain diseases to make cures. We're working on that too in our own way, doing fundamental work. We're working with Li Wei Tsai at MIT, who's head of the MIT's Picower Institute, and I have a fantastic postdoc, Alice Stanton, trying to create a brain on a chip with 6 different cell types and a matrix and blood-brain barrier that could mimic this. And I hope through things like that or what other people do, we'll be able to someday come up with targets for those too, or somebody. But the problem is for Parkinson's and for Alzheimer's, I just don't think the biology, this is my 2 cents, is well understood. So you could make RNA, but you wouldn't know what to make it to do.
Just very quickly, is it to do with the material of the neurons and the brain, or is it the other side? Like,
And I think that's the point. There's different theories. People have theories about, you know, amyloid, about tau. There's all different kinds of theories. Maybe some of them are right or partially right. But, you know, I could tell you that that I'm hardly any expert, but the neuroscience community has hotly debated these things for many, many years.
Yeah, and I'll just remind you guys, if whoever was at Melanie Iverson's, you know, Science Cafe, she literally said the sequence of the virus was published on the internet, and they literally typed in the sequence of the spike protein with the appropriate modifications that they knew of from an NIH investigator. That then made the mRNA that got put into a lipid nanoparticle. So the manufacturing cost is also matched now by manufacturing and development speed. So things are happening faster. So maybe we won't have to guess at.
Yeah, but you'll need to understand the biology. That has been, because I think you first have to do that. And that's the beauty of the things you said. Enough technologies had been developed so that biology was understood. Whereas sadly, I mean, someday you're right. And that actually ties the 2 questions together. You know, I think the manufacturing things are there, but I think this is why curiosity-driven science is so important, because someday somebody will figure out advances, you know, more basic understanding of cancer, of brain diseases and so forth. And why new tools, like there's a brain on a chip or other things, might help, but it's a big challenge.
We have a question for this young person.
Oh, well. Go ahead.
What gave you the drive to keep going even after you got rejected many times?
Yeah, well, it's a good question. Sometimes I ask myself that question. But I believed, you know, I could see when I did some of these things, because with my own eyes, that molecules were coming out of the little particles. I could also see those blood vessels being stopped. So I just felt it was important, and I felt if we could do this, it would someday make a big difference. I didn't agree with the people who told me I was wrong. You know, like I say, and I'm pretty stubborn. But so I think it's a combination of a lot of things. I was also, I think, very fortunate to have done postdoctoral work with Judah Folkman, who I mentioned. He got criticized a lot, and I got to see him as a role model for me, that he, you know, kept, even though, because he had this idea about the blood vessels, and people criticized him. I mean, you know, so a lot of scientists and scientific journals criticized him and said this was, you know, this was the whole thing was fiction in the mind of the scientist, and he kept going. So I think seeing him and his example also made a big difference to me.
So I'm just gonna take one question from the online group, and I'm gonna modify it slightly. Just, do you think, you know, or because of what you focus on, like how would we define the field that you've created now? That this is no longer a simply, like we took chemistry, biology, or physics. Like this is now something that you need to have a, kind of eye of newt, wing of bat. You need to have a cauldron of understanding. And how do we train our young scientists in that vein?
Yeah, well, I think there's different things embedded in your question. I mean, in terms of calling the field, people have called me all kinds of things. But sometimes it's modern biomaterials. Sometimes it's macromolecular drug delivery. Tissue engineering, all those have been attributed to, I suppose, to what we've done. But how you train people, I still think it depends at what stage. I still think that if you're a student, the most important thing to me is learning fundamentals of chemistry, biology, engineering. And then as you go further, I do think it's the kind of training in a lab where you learn how to address problems. I mean, I certainly never dreamed that things I just didn't know. But you try to solve problems. I mean, one of the great things to me about an engineering education is it teaches you about solving problems.
Do you see any applications of some of this technology in autoimmune disorders coming this way?
Yeah, well, I think that's a great question. And I do, but I do feel that the problem with autoimmune diseases is not unlike what I mentioned for brain diseases. I don't think that the biology is well enough understood. I think, we've certainly talked about that. And I think the types of tools that we're developing and that others have developed can help those. But I think until they're better understood, that's still the biggest problem, is not understanding the fundamental pathology, the fundamental biology well enough. I mean, like I said, I'm saying that not as a biologist or a clinician, but that's my concern. I'd love to see us be in the position to do that. I think the thing, when you look at a lot of what Moderna or Nylam or other companies, Genentech, that I've been involved in have done, is they've gone after diseases, I mean, sometimes they've figured out the biology a little bit more, just like Dr. Folkman and I did with the angiogenesis work. But I think you have to know that before these things can be used really effectively.
Yeah, but I will say, I think some of these innovations, again, allow you to start testing those ideas that you can even faster. So I think, we won't always get it right, but we'll get to do those clinical trials faster from the conception to the test. And so I think that, we're going to find ourselves seeing much more innovation faster in this field. Okay, one more question. Is there?
Just, how old were you when you read your first article in Scientific American?
How old was I when I read my first book? Boy, I'm guessing probably fairly old. I'm sure I was in my 20s. I read Tom Swift books. that when I was a little boy. I like those. But I don't know when I really, I don't know that I understood science well enough to really have appreciated all that Scientific American stuff. I've now written probably 4 or 5 articles for Scientific American, but I hope people read them. But I'm sure I was in my 20s.
Well, thank you so much, Bob. I think this has been remarkable for everybody. And, use it to follow your nose. I don't know. That's one of the take homes here. I think the ability to be able to have, we'll call it perseverance, not stubborn, curiosity, and the ability to be able to have that inner conviction and courage to follow it. And I think Bob has that in spades. So thank you again for him. And thank you guys for all being here. And thank you online. Thank you.